Usually when we talk about a bird flu outbreak making the headlines, there is this immediate collective gasp. We instantly picture the absolute worst case scenario.
Right, it's that knee-jerk reaction.
Exactly. It feels like this terrifying, inevitable math equation. A bird gets sick, a human catches it from the bird, that human gives it to another human, and suddenly, bam, we are looking at a global pandemic.
And honestly, given the historical devastation of pandemics, that fear is entirely understandable. I mean, the math seems pretty straightforward on the surface.
But then you actually look at the epidemiological history of avian influenza, and that simple math equation just, doesn't add up. Every avian flu doesn't automatically jump to humans.
No, not at all.
In fact, the vast majority of them hit a wall, which raises this massive fundamental question for us today. What exactly is that wall? Like, what is the hidden microscopic barrier inside our cells keeping us safe from these zoonotic spillover events every single day?
Yeah, it really is the absolute definition of a biological firewall. I mean, for a long time, immunology and virology understood there was a species barrier, but we didn't fully understand the molecular bricks and mortar of that wall, if you will.
Right, we just knew it was there.
Exactly. We knew humans were generally resistant, but the precise cellular mechanics of that resistance remained largely a black box.
Well, today's deep dive is blowing that box wide open, and the implications are just incredibly exciting. We are looking at a groundbreaking 2026 research paper from the journal Science.
A truly phenomenal piece of work.
It is. The researchers discovered a highly specific molecular alarm system inside our cells. It's called the Cysteine NF-EGAD34 pathway, and it physically protects us from bird flu.
And, rather terrifyingly, they also found the exact evolutionary lockpick that human-adapted flus use to bypass that system.
Yeah, that's the scary part. So our mission today for you listening is to unpack the mechanisms, the molecules, and the single tiny mutation highlighted in this paper. We're going to explain every piece of this puzzle.
Right, and the density of the information in this study is remarkable. So to keep it clear, we're going to break this down into 3 major chapters. First, we'll explore the architecture of the cellular alarm itself.
The alarm, got it.
Then we'll look at the specific defensive protein, the enforcer that actually neutralizes the virus. And finally, the viral lockpick, the countermeasure.
Perfect. And we'll synthesize the key takeaways with little recaps as we move through each phase so the overarching narrative of this microscopic arms race remains crystal clear for you.
Sounds like a solid plan.
Okay, so before we even get into the mechanics of the alarm, I have to point out what looks like a massive biological paradox right out of the gate.
Oh, the DNA versus RNA issue.
Exactly. Influenza A, the flu, is an RNA virus, right? Its genetic code is made of RNA. But STING, which stands for stimulator of interferon genes, is famously known in immunology as a DNA sensor.
Right. Its primary job is to detect misplaced double-stranded DNA floating in the cytosol,
Which is usually a sign of a bacterial infection or a DNA virus. So why on earth is an RNA virus tripping a DNA alarm system?
That paradox is exactly what catalyzed this entire line of research. I mean, for years, the prevailing assumption was that because the flu is an RNA virus, it must be fought exclusively by the cell's dedicated RNA sensors.
Like the RLR pathway, right?
Yeah, exactly. The REGI-like receptor pathway. But researchers started noticing this massive discrepancy in the data. Even when they experimentally knocked out those RNA sensors in cellular models, the cells could still mount a robust defense against the flu.
Wait, really? With no RNA sensors at all?
None. But here is the kicker. When they removed S, the DNA sensor, the influenza virus, replicated completely out of control.
Okay, so the RNA virus was somehow activating the DNA sensor. But, I mean, if there's no DNA intruder to physically bind to STING, what is flipping the switch?
Well, the researchers observed that STING was getting phosphorylated during the flu infection.
Phosphorylated, meaning it gets a chemical tag added to it.
Yes, specifically at a site called serine 366. That phosphorylation event essentially forces STING into its active conformation. The virus's mere presence, maybe through cellular stress or some intermediate signaling, was manually tripping the alarm.
Wow. Okay, so the DNA alarm is definitely ringing despite the lack of DNA. But here is where the pathway gets super weird.
It does get a bit unconventional here.
Right, because when STING normally detects DNA, it triggers a cascade called the IRF3 pathway, which leads to the release of interferons. That's its classic textbook function.
The standard immune response, yeah.
But this paper shows that against the flu, S completely ignores IRF3. It uses a secondary independent pathway called NFEB. How does a single sensor protein know to route the signal to a completely different defense network?
It all comes down to the structural architecture of the S-sting protein itself. To answer that question, the researchers essentially performed a massive genetic audit.
A genetic audit? What do you mean?
They took the S-sting protein and systematically screened 90 different point mutations. They were looking for the precise physical switch, the exact amino acid that controls this secondary NFV pathway without disrupting the primary IRF3 pathway.
And they found it, right? At amino acid residue glycine 90.
Yes, Gly90. Physically, this amino acid sits in a very specific structural hinge on the protein called the TM2-TM3 linker region.
Which acts as a bridge between two sections of the protein.
Exactly. It connects two transmembrane sections. And the researchers discovered that if you mutate that specific glycine and replace it with an alanine, creating a mutation known as G90A, the S-sting protein undergoes this really subtle structural shift.
Let me guess. That tiny shift severs the connection to the secondary alarm.
It absolutely severs it, yes. With that G90A mutation, S-sting can still trigger the normal IRF3 interferon pathway perfectly fine. It still acts as a DNA sensor.
But its ability to trigger the NFE alarm drops to zero.
Absolutely to zero.
Mechanistically, why does that one single substitution break the circuit?
Because to turn on NF key, the STING protein needs to physically bind to a downstream adapter protein called TREF6.
TREF6, okay.
Right, and Gly90 is crucial for maintaining the precise 3D shape required for that docking mechanism. When you change that glycine to an alanine, STING loses its ability to grip TREF6. The structural lock no longer fits the key, and the NF key circuit is totally broken.
Which brings up this fascinating evolutionary context from the paper. They didn't just look at human STING, they looked at STING homologs in really primitive animals.
Yeah, the comparative immunology in this paper is amazing.
They examined the sea anemone, Nematostella victensis, and the tropical frog, Xenopus tropicalis. And those older evolutionary versions of STING, they don't have this NF key activation ability at all.
They don't. They can mount the standard interferon response, but they cannot route the signal through TREF6 to NF key.
Meaning, this entire secondary alarm system is a relatively recent mammalian evolutionary upgrade. We evolved this specific structural hinge, this Gly90 switch, to deal with a specific set of threats that amphibians and invertebrates just don't face in the same way.
And we desperately rely on that evolutionary upgrade. The researchers demonstrated this with a rather striking in vivo model.
The mouse model, right?
Yes. They genetically engineered mice to possess that exact G90A mutation. Now, they didn't knock out a STING entirely. They just made that one tiny substitution to break the NFU connection.
And what happened when they infected them with the flu?
When they infected those mice with influenza A, the results were catastrophic. The mice suffered massive alveolar structural damage in their lungs and exhibited just astronomically high viral loads.
Because without that specific Gly90 switch turning on the NFU pathway, the primary interferon response simply wasn't enough. They were virtually defenseless against the flu.
It really highlights the necessity of redundant, specialized biological systems.
Okay, so we're at our first checkpoint. Let's do a quick mini recap for everyone listening.
Good idea.
Think about your own home security system. If the front door sensor breaks, does the window sensor still work? STING proves our cells have backup wiring. So, bullet 1, STING fights RNA flea viruses, even though it's a DNA sensor.
Bullet 2, it uses a secondary alarm pathway called NFT to do it.
And bullet 3, the Gly90 amino acid is the physical switch that turns the specific alarm on.
Perfect summary.
But that logically leads us to the next major phase of this mechanism. Chapter 2. STING trips the Gly90 switch. The NFE alarm sounds. But an alarm is just a signal, it's just noise.
An alarm doesn't stop a burglar.
Exactly, the police do.
Yeah.
If the virus is replicating rapidly inside the lung tissue, what is the actual physical weapon the cell deploys in response to that alarm? Who shows up to fight the virus?
Well, the NFT pathway essentially acts as a master transcription factor. When it gets activated by a STING, it translocates to the nucleus and starts aggressively upregulating a specific set of genes.
The paper refers to these as NSGs, right? NF-stimulated genes.
That's right. And the researchers conducted a comprehensive screening of these NSGs to determine which one was actually responsible for inhibiting viral replication. And the standout protein, the undisputed heavyweight in this antiviral response, is called GADD34.
GADD34? Growth arrest and DNA damage-inducible protein 34. Now, wait a minute. Because this is where the biology seems completely backwards to me.
You're talking about its day job.
Yes, because GADD34 is a well-documented phosphatase. During a cellular crisis, like when a cell is starving or under attack, protein translation usually shuts down to conserve energy. GADD34's normal cellular job is to remove chemical tags and restore that protein translation.
It acts like a factory worker getting the assembly lines running again.
Exactly. It gets the cellular factories running. But if a virus is trying to hijack the cell's ribosomes to build more of itself, why would the cell deploy a protein whose primary function is to turn the manufacturing floor back on? Wouldn't that actively help the virus?
That is the exact paradox the researchers had to untangle. And it turns out GADD34 is moonlighting.
Moonlighting, like it has a second job.
Exactly. During an influenza infection, it largely abandons its role as a translation-restoring repairman and takes on a highly targeted antiviral function. It becomes a bouncer.
Okay, but how did they prove it wasn't just using its normal phosphatase tools to somehow fight the virus?
By mutating the specific domains of GADD34 that control its phosphatase activity, they introduced mutations known as R591A and CARA, effectively neutralizing its ability to restore protein translation.
So they completely disabled its normal enzymatic tool.
Yes, and even with those tools disabled, GADD34 still stopped the influenza virus cold. Its day job genuinely has nothing to do with its antiviral power.
That is wild. So what part of the protein is actually doing the fighting, if not the phosphatase part?
A completely different structural region called the PACE2 domain.
Okay, so let's unpack that. How does this PS domain physically stop an RNA virus from multiplying?
Well, to replicate its genetic material, the influenza virus relies on a highly specialized piece of machinery called the viral ribonucleoprotein complex, or VRNP.
You can think of it as a viral 3D printer, right?
A very good analogy.
Yeah.
Yes, a 3D printer consisting of several crucial polymerase proteins, specifically PB1, PB2, and PA. The piece domain of GADD34 acts as a targeted molecular wrench thrown directly into the gears of this printer.
Oh, wow.
It physically binds to those individual viral polymerase components and just jams the machinery.
It physically prevents the virus from copying its genome.
Precisely. The virus manages to enter the cell, sure, but its replication apparatus is structurally paralyzed by GADD34. It is effectively sterilized.
And the clinical application of this mechanism in the paper is absolutely staggering. I mean, to test the sheer potency of GADD34, the researchers utilized AAV adeno-associated virus as a gene therapy vector.
This was a really elegant experiment.
It was. They used mice that were genetically modified to lack the GADD34 gene entirely. Normally, these knockout mice succumb rapidly to even a minute sublethal dose of the flu. Their lungs just can't clear it.
They have no enforcer.
Right. But the researchers used the AAV vector to preemptively deliver human GADD34 directly into the respiratory tract of these highly vulnerable mice.
They were essentially arming the cells with the weapon before the invasion even occurred.
And the results speak for themselves. After vectoring in the human GADD34, they exposed these mice to highly lethal doses of influenza, titers that would normally guarantee a 100% mortality rate.
Oh, yeah.
The prophylactically administered GADD34 dramatically suppressed viral replication. It significantly reduced alveolar pathology, and the mice survived. By just providing that one specific protein, they rendered previously defenseless mice functionally immune to severe disease.
Which is incredible. When you scale that up, it demonstrates that GAD34 isn't just an interesting downstream effector. It is a potent, standalone antiviral mechanism that our respiratory epithelial cells naturally deploy.
Okay, so that brings us to the 10-minute mark. Let's synthesize the second chapter for the listener.
The enforcer phase.
Right. Bullet 1, NFG activates GADD34. Bullet 2, GADD34 abandons its day job as a phosphatase to fight the virus.
In bullet 3, it uses its PS domain to jam the viral copy machine, the VRMP complex.
Exactly. But this leaves a glaring, almost existential question for you listening. If our cells have evolved such an incredibly effective bouncer in the lungs, and this sting to AD34 pathway is so robust, why do human beings still get the flu every single year?
It's the multi-million-dollar question.
Right. I mean, if the firewall is this good, what is the virus doing to get past GADD34?
Well, welcome to Chapter 3, the lockpick. It is the classic evolutionary arms race. Viruses are the ultimate lockpicks. They are constantly mutating, probing our security systems for vulnerabilities.
And human flu found one.
It did. The researchers discovered that human-adapted influenza A has evolved a brilliant, highly specific countermeasure. It uses a viral structural component called the matrix-1 protein, or M1, to silence the S sting alarm before GADD34 ever gets the signal to deploy.
So M1 acts as the burglar, literally cutting the alarm wire.
That's a perfect way to visualize it.
Mechanistically, though, how does a viral matrix protein shut down a mammalian DNA sensor?
By direct physical interference, the M1 protein binds directly to the SANG protein. Specifically, it attaches to a region on S-sting spanning residues 317 to 343.
Which is physically distant from the Gly90 switch we discussed earlier.
Very distant, yes. But by docking at the specific site, the M1 protein creates steric hindrance.
Meaning it just gets in the way.
Exactly. Physically blocks STING from interacting with a crucial downstream kinase called ICO.
And without ICO,
the signal cannot propagate. The NFD alarm never sounds. Since the alarm doesn't sound, the cell doesn't transcribe GADD34. The wrench never gets thrown into the viral 3D printer, and the virus replicates unchecked.
Okay, here is where the paper delivers its core insight regarding zoonotic spillover. It definitively explains the molecular barrier between birds and humans.
The crux of the whole paper.
Exactly. And for the listener, I want you to use a simple mnemonic to remember the exact amino acid difference that defines whether a flu stays in birds or causes a human pandemic. It all comes down to one single evolutionary substitution in that viral M1 protein, residue 115.
The magic number.
Right. Here is the mnemonic. Avian flus predominantly have valine at position 115. Think V for vulnerable.
V for vulnerable, I like that.
And human flus have isoleucine at position 115. Think I for invader or invincible.
The V versus the I. It is an incredibly precise distinction. And when the researchers analyzed decades of sequence data, the evolutionary divergence was just stark.
What did the numbers look like?
Over 95% of all human pandemic and seasonal flu strains possess that isoleucine the I at position 115. Conversely, when they examined wild avian flu sequences, only 0.31% carry that specific mutation. The overwhelming majority of bird flus have the valine.
Okay, but valine and isoleucine are structurally very similar amino acids. They're both hydrophobic, but isoleucine has just one extra carbon atom, right? Just a tiny bit of extra bulk.
A minuscule difference, structurally speaking.
How does that microscopic difference completely dictate whether the lockpick works on a human cell?
It alters the docking surface just enough to matter. The avian M1 protein, with its valine at 115, is perfectly adapted to bind to avian sting. It blocks the alarm in birds efficiently.
But mammalian sting has evolved differently.
Right. So the avian M1 protein is literally the wrong shape for our locks. It cannot bind to human sting. Therefore, when an avian flu virus enters a human lung cell, it is unable to block our sting protein.
So our NFDB alarm sounds.
The alarm sounds. GADD34 is rapidly synthesized, the viral replication machinery is jammed, and the bird flu is destroyed. The virus is, as your mnemonic suggests, vulnerable.
But the threat emerges when that virus mutates.
Exactly. If that avian flu undergoes a chance mutation, swapping that valine for an isoleucine, creating the human-adapted I protein, the 3D folding changes just enough.
And suddenly it fits the lock.
Yes. Yeah. It gains the ability to successfully dock with human sting at residues 317 to 343. It blocks ICO, silences our alarm, and keeps GADD34 dormant. The flu then freely replicates and spreads from human to human.
Unbelievable.
That single amino acid substitution at position 115 is the physical key required to unlock a spillover pandemic.
It is genuinely awe-inspiring when you scale that up. So let's do our final quick summary here for Chapter 3.
Go for it.
The viral M1 protein attempts to bind to sting to disable the cellular alarm. The success of this binding, and thus the difference between a harmless, dead-end bird flu infection and a proliferating human pandemic, hinges entirely on amino acid 115.
On that M1 protein, yes.
Right. V, or valine, means that the virus fails to block human sting and is destroyed. I, or isoleucine, means it successfully evades the alarm and invades the host. Think about how fragile a pandemic really is.
Incredibly fragile.
A single amino acid change, one microscopic Lego out of millions, is the absolute difference between a virus fizzling out in a single isolated case and a virus acquiring the capacity to sweep across the globe.
It reduces the terrifying complexity of a pandemic down to a highly predictable molecular interaction. And this brings us to the broader implications of why this 2026 science paper is such a pivotal development.
It's a total game changer for surveillance, isn't it?
It absolutely is. It transcends basic biology. It provides us with a brand new, highly targeted molecular marker for global health surveillance. For years, epidemiology has monitored the threat of bird flu by looking at larger macroscopic factors, or broad receptor binding profiles.
Which are useful, but maybe not as precise.
Right, but now we have a highly specific molecular radar. By continuously sequencing and surveying wild avian flu populations for that precise valine to isoleucine mutation in M1115, scientists can theoretically predict with mathematical precision which emerging strains are on the verge of developing the capacity for human-to-human transmission.
We can essentially monitor the virus as it attempts to fashion the lockpick in real time.
Exactly. Furthermore, understanding this exact mechanism opens up a massive new frontier for targeted therapeutics.
Oh, like new drugs.
Yes, if pharmacologists can design small molecule drugs that physically occupy that binding site on humanist sting, essentially putting tape over the lock so the viral M1 protein cannot attach them, we could artificially keep the alarm system online.
That is brilliant. It would let GADD34 do its job.
Exactly. That could represent our next major antiviral weapon, not just against seasonal flu, but against novel pandemic strains.
So we began this deep dive discussing that instinctual fear of a bird flu headline, right? And the invisible biological wall that stands between us and a pandemic. And now we have a blueprint of that wall.
A very detailed blueprint.
It is an intricate, relentless, microscopic arms race occurring within our respiratory epithelium every single day. We possess a repurposed sensor in Esting, a heavy-hitting moonlighting enforcer in GADD34, and we face an ever-evolving viral lockpick in the M1 protein.
It really is a marvel of biological engineering. Multiple overlapping systems, structural redundancies, and constant evolutionary pressure, all interacting at the atomic level to maintain equilibrium.
It serves as a profound reminder of how intricately adapted our immune systems truly are, which leaves us with a provocative final thought for you to carry with you today.
Let's hear it.
We've just examined how a single virus utilizes one microscopic mutation to bypass millions of years of targeted mammalian immune evolution. So if viruses are constantly probing our cellular alarm systems, searching for that one subtle structural advantage, what other deeply hidden, unknown defensive pathways are currently quietly protecting you right now?
That is a fascinating and admittedly sobering question.
And what other viruses are out there silently iterating, trying to learn the code to disarm them? Keep that in mind next time you see a headline.